Welcome all to the today's session. So today we continue with the genetics module. Last session we have finished with the mitosis. This week we are going to finish with the meiosis, which is a reduction division. So there is a difference between the mitosis and meiosis. So mitosis happens in the cells which is happening in the
But I'm not able to hear you. Hello.
okay. Now are you able to hear? Is it okay now?
Yes,
Is it
sir.
okay?
Yes, sir. Yes, sir.
Yeah.
Yes, sir.
Good evening, all. We begin with today's session. We begin with our quote, Master MRCOG with concept-oriented clarity. So we are in the genetics module. In the last session, we have finished with the mitosis. Okay. So this now session will deal with meiosis. Then we will discuss about the chromosome structure, pedigree analysis, chromosome aberration, etc. So we continue with the meiosis now. So the purpose of the meiosis is the growth and repair. Why purpose of the mitosis is mainly the growth and repair, while meiosis is concerned with the gamete formation. So meiosis is seen mainly in the germ cells, while mitosis is seen in the somatic cells. So what happens in the mitosis? There is only one division, while in meiosis there are two divisions. First is a reduction division, and second division is a equational division, like what happens in mitosis. Then in mitosis only two daughter cells are formed. In meiosis, four daughter cells are formed. So chromosome number after the mitosis will remain same, that is diploid cells you are going to get, while in meiosis you will get the haploid cells. Then what will be the genetic makeup? In mitosis, genetic makeup will be identical, while in meiosis there is going to be variation in the genetic content. Okay, this is what is the key feature of the meiosis, and there is always one question either on mitosis or meiosis, mainly on the meiosis. Okay, so mitosis is concerned with the mainly genetic stability because the same genetic makeup is there, while meiosis there is a genetic diversity. So same thing is shown here, that from this mitosis which is concerned with the growth and development, and you get two cells which are diploid in nature at the end. Okay, here you get a two cells, while in meiosis it is a reduction division. There are two divisions are involved. First there is a haploid division, and finally you get four haploid cells. Okay, so each cell will be haploid with 23 chromosomes, and it happens in the gametes. Okay, so mitosis versus meiosis comparison we have just finished. Now let us see the basic concept of meiosis. So it happens in the specialized cell. It's a specialized cell division to produce gametes, that is sperm and ova. It converts diploid cell, that is 2n, into haploid n cell. Okay, so it maintains the chromosome number across the generation. Fertilization restores the diploid state. So once there is a fertilization between the two haploid cell, there will be restoration of the diploid state. Okay, so in humans, uh, we know that we have 46 chromosome, that is equal to 2n. So it will give you 23 chromosomes in each gametes. Okay, so key features of meiosis is there are two sequential division. The first division is a meiosis I, which is a reductional division. Second division is a meiosis II, that is a equational division. That means there is no reduction in the further reduction in the chromosomes. Okay, so separation, that is meiosis I, is concerned with the homologous chromosome separation, while meiosis II is concerned with the sister chromatid separation. Okay, so final outcome of meiosis is a four haploid genetically unique cells. So coming to the phases of meiosis, each division, that is meiosis I and meiosis II, is divided into four phases, that is prophase, metaphase, anaphase, and telophase. So we will begin with meiosis I first, which is a reduction division. Okay, so before the meiosis I begins, there is a small phase called as interphase. Okay, that happens before meiosis I only. There is no interphase uh, between the meiosis I and meiosis II. Please remember. Okay, so there is an interphase before meiosis I where there is a DNA replication which happens. So it is further divided into G1 where there is a growth, then S phase where there is a DNA synthesis, and G2 phase where the cell is prepared for the division. Okay, so the sub stages of this meiosis I are prophase I. So what happens in prophase I? It is there are unique features which are, or unique events which are happening in the prophase I. There is a synapsis. That is the homologous chromosomes will pair to each other. Then there is a formation of the bivalent, that is tetrad will get formed. Please remember these words because these are the questions which are asked, especially on the features. The tetrad is formed in which stage? Okay, so it is the meiosis. Meiosis one, prophase one, prophase one of the meiosis. So first is a synapsis where the homologous chromosome will pair with each other. There is a formation of the bivalent leading to the tetrad formation. Then there is a crossing over, that is the exchange of genetic material between the homologous chromosomes. It occurs via synaptosomal complex. Okay. Then there is a chiasmata formation, that is the visible crossover points. Okay, this is also, you should know what is chiasmata. These are the visible crossover points. Then it maintains the homologous chromosome attachment. What are the key facts here? At least one crossover per chromosome pair is seen. However, there can be more than one crossover also. Okay, see this particular figure here in prophase. These are the homologous chromosomes which are paired now. This is also one pair. This is also one pair. Okay, and this is, you see, this is the chiasmata. So here there will be crossing over between these two. Here in one chromosome only you are getting two chiasmatas. So there can be crossing over at multiple points also. This is the prophase one of the meiosis. So the chromosomes condense and the nuclear envelope breaks down and there is a crossing over. Okay, so mnemonics, this prophase one of meiosis one is further divided into substages, that is liptotin, zygotin, pachitin, diplotin, and diakinesis. Okay, so there is a mnemonic to remember: le zi pa didi. Okay, le zi pa didi. If you remember, that is leptotin, zygotin, pachitin, diplotin, and diakinesis. These are the substages of prophase one of meiosis one. Okay, so leptotin. What happens in the leptotin stage of prophase one is there are thin threads are formed. The chromosomes will begin to condense. They appear as a long, thin, thread-like structures. Each chromosome already consists of two sister chromatids, not yet visibly separate. Okay, so chromosomes become visible for the first time in this particular phase, that is the liptotin. Okay, so then there is a second stage, that is the zygotin. What happens in the zygotin? That there the synapsis begins. Okay, this is what we have seen in the beginning as an overview of prophase. So now in zygotiny, the synapsis begins. The homologous chromosomes will pair precisely. There is a formation of bivalents. Okay, so two chromosomes will come close to each other for leading to formation of tetrads. Okay, so each chromosome will have two sister chromatids and you will be able to see the tetrads. Okay, mediated by the synaptosomal complex. Okay, so key point here to remember is the zygotin stage is the homologous pairing is initiated. So when you are asked about the homologous pairing, it is the zygotin substage of prophase 1 of meiosis 1. Okay, it is the homologous pairing which gets initiated where you get tetrads. They can ask you, where do you see the tetrads? It is the zygotin stage of prophase 1 of meiosis 1. Okay, so that is how you see the this second stage. Then third stage is a Pachitini stage. That is a thick stage, you can remember. Chromosomes will become shorter and thicker. Then there is a crossing over which happens in this Pachitini stage. So exchange of genetic material between the homologous chromosomes and formation of recombinant chromosomes. So there is an exchange of genetic material in this Pachitini stage and synaptosomal complex is fully formed here. Okay, synaptosomal complex is fully formed. Okay, the next stage is the diplotiny stage. Here synaptosomal complex dissolves, homologous chromosomes will begin to separate, and most important here is they remain attached to the chiasmata. Okay, that is the site of crossing over. And in female oocytes, oocytes are arrested in the diplotiny stage until the puberty. So this is one of the question which they ask you. So what is the stage at which these oocytes are arrested during the in that embryonic stage? So they are arrested in the diplotiny stage until the puberty. Okay, so this is the stage you should remember from this particular SBA point of view. So in diploteny, there are synaptosomal complex will dissolve, homologous chromosomes will begin to separate, they will remain attached to the chiasmata, that is the site of crossing over, and most important clinically in females, the oocytes will be arrested in the diploteny stage until the puberty. The last stage is diakinesis. So here in diakinesis, chromosomes are maximally condensed, they are clearly visible, and chiasmata terminalization, that is they move towards the end. There is a nuclear membrane will break down and spindle formation will begin. Okay, so this prepares the cell for metaphase 1. So here the prophase 1 gets finished and then the metaphase 1 will begin. So prophase was divided into further substages, which we have just seen. Okay, then prophase 1 will be followed by the metaphase 1. So prophase 1 is most commonly asked. Okay, then metaphase 1. Okay, so this is the just overview of this prophase 1 is mentioned here. So in leptoteny stage, there is a chromosome condensation, first visible stage. In zygoteny stage, there is a synapsis, bivalent formation or the tetrad formation is there. In pachyteny stage, there is a crossing over, there is a genetic recombination which happens. In diploteny stage, there is a chiasmata which is visible and the oocyte is arrested in this particular stage. Then there is a diakinesis where there is a terminalization and preparation for the metaphase. Okay, so just to remember, crossing over pachyteny stage, synapsis zygoteny stage, chiasmata visible diploteny stage, oocyte arrest in the diploteny stage, and most tested stage is a pachyteny stage. Okay, so that also you should remember. Okay, now coming to the metaphase 1. In metaphase 1, homologous pair will align at the equator. There is a random orientation, independent assignment. Independent assortment. Okay. Most important here is a chromosomes attached to the opposite spindle poles. So in metaphase 1, what you see is the homologous pairs will align at equator. There is a random orientation. This is the chromosomes are arranged at the equator. Okay, in the metaphase 1. So random orientation is there and chromosomes attached to the opposite spindle poles. So here you will see these chromosomes are attached through the spindle fibers to the opposite poles. Okay, so this is the metaphase 1. So pairs of homologous chromosomes move to the equator of the cell. Okay, that is the metaphase 1. Then in next phase is anaphase. So homologous chromosomes will separate, sister chromatid will remain attached. So result is a reduction in the chromosome number, that is from 2n to n. Okay, so in anaphase 1, the homologous chromosomes will separate. So this is the anaphase 1 where the chromosomes have started separating from each other. Okay, so homologous chromosomes will move to the opposite poles of the cell. So next stage is a telophase 1 and followed by the cytokinesis. So in telophase 1, two haploid cells are formed. Chromosomes are still duplicated. Okay. So this is the telophase 1. Then there is a cytokinesis. Chromosomes gather at the opposite poles of the cells and the cytoplasm divides. That is the cytokinesis. Okay. This is the meiosis 1, which happens. After the meiosis 1, there is a meiosis 2. This meiosis 2 is same as like a mitosis. Okay. The only difference is that in meiosis 1 and meiosis 2 is there is no DNA replication before this stage. This I have already mentioned when we started with meiosis 1. Okay, now you will have same stages, prophase 2, metaphase 2, anaphase 2, and telophase 2. Okay, so the same stages will be there. Now let us see about the stages of meiosis 2. So first will be prophase 2. Here the chromosomes will condense, nuclear membrane will break down, and spindles will get formed. In metaphase 2, chromosomes will align at equator, spindle fibers attached to the opposite poles. Okay, so you have prophase 2. Here in metaphase 2, the spindle fibers are chromosomes are attached at the equator. Spindle fibers are formed. Okay, in metaphase 2. Then anaphase 2, their sister chromatids will start separating. So in anaphase 2, there is a separation of the sister chromatids. Okay. And in telophase 2, followed by the cytokinesis, there is a formation of the 4 haploid cells. Each chromosome has a single chromatid. Okay. So the purpose of meiosis is the genetic variation. This we should remember. Okay. So how does this happen? This happens by crossing over. There is an exchange of alleles between the homologous chromosomes that produces the recombinant chromosomes. And there is an independent assortment. So random alignment of homologous in metaphase 1. In humans, possible combination is 2 2 is to square of 23 equal to 8 million gametes. Okay. So from key examination point of view, meiosis 1 is a reduction division. Meiosis 2 is mitosis-like division. Crossing over happens in prophase 1. Chiasmata is the site of genetic content. Then independent assortment is a genetic leads to the genetic diversity. And the final product is the 4 haploid non-identical cells. If there is any error, that will lead to the aneuploidy. Okay, non-disjunction. Okay, so if the chromosomes fail to separate, that will lead to non-disjunction. This we should understand because there can be question on this. Okay, so it will lead to the trisomy 21 or monosomy X, that is a Turner syndrome. So in meiosis, what happens? There are 2 divisions. Okay, so just for the understanding point of view, we are taking one pair. Okay, so in first reduction division, so this is the one chromosome goes here, another chromosome goes here. Okay, and in second is a mitotic division. So one chromatid will go here, one chromatid will go here, and similarly this should happen. However, in non-disjunction, which can happen in the first division or it can happen in the second division. Now let us see what happens in the non-disjunction. The principles of meiosis and the non-disjunction. For simplicity, one pair of newly replicated autosome is shown here. So now here out of one pair, that is 2 chromosomes, here there is a non-disjunction. One pair does not get any chromosome, and both the chromosomes are going into the one cell, and which is then further dividing, and each becomes the disomic, while this is nullisomic and this is the disomic. Okay, so the wrong gametes are formed here. Here what is happening now? Here this is the pair which we are thinking of. Here the first division is quite good. Okay, so here appropriately there is a genetic material is getting divided. There is a uh, reduction division. The first reduction division is proper. However, in the second reduction division, it is not proper. And here, one cell is not getting any genetic material, while all the genetic material goes to other cell. Okay, and here again the reduction division. Here again this mitosis or the meiosis 2 is good, proper. So it leads to the nullisomic and disomic gametes. Okay, so this is called as non-disjunction. Okay. So meiosis produces 4 genetically diverse haploid gametes through reduction division, crossing over, and independent assortment. So these are the mechanisms by which genetic diversity happens. One is reduction division, second is a crossing over, and third is an independent assortment. So these are the things which are involved in the meiosis from examination point of view. We have certain questions, doctor. Can you please read the question? We can solve some questions.
Yes, sir.
Yeah. Yes, go ahead.
A primary oocyte is undergoing meiosis, during which stage does crossing over between homologous chromosome occurs. So prophase 1.
So crossing over is happen in prophase 1. So crossing over occurs in prophase 1, that is Pachytiny stage, substage of prophase 1. It involves the exchange of genetic material between the homologous chromosomes responsible for genetic recombination. Next question, doctor.
Which stage of meiosis is responsible for reducing the chromosome number from diploid to haploid? Anaphase 1, telophase 2, anaphase 2, telophase 2, anaphase 1, prophase 2, metaphase 2. Sir, option C, anaphase 2. Anaphase 1, sir.
Anaphase 1. Yeah,
Anaphase 1.
anaphase 1. Okay. So homologous chromosomes separate in anaphase 1. That leads to reduction division from 2n to n. Okay. Next.
Independent assortment of chromosomes occurs due to separation of sister chromatids in anaphase 2, random alignment of homologous chromosomes in metaphase 1, DNA replication in S phase cytokinesis after telophase 2, crossing over in prophase 2. So
It should be,
random alignment of
yes.
homologous chromosome in metaphase 1.
Yeah, it's B. Okay, random alignment of homologous chromosomes in metaphase 1. So orientation of homologous pairs is random at metaphase plate. It leads to the genetic diversity, gives rise to 8 million combinations in humans. Next question, doctor.
Which of the following best describes meiosis II? Reduction division, separation of homologous chromosome occurs after DNA replication, separation of sister chromatids produces diploid cells. Meiosis II, no? Separation of
So is it C occurs after DNA replication?
No, it is meiosis 2. No, what happens in the meiosis 2? There will be separation of
it's
the
like
sister.
mitosis. So
Yeah,
option D.
it should be option D. Okay, let us see. Yeah, separation of sister chromatid. Because in meiosis 1, chromosomes will separate. In this meiosis 2, each chromosome, sister chromatid will get separated. Okay, because we are concerned with the 2n first. 2n will lead to n. And from n, what we are going to get? Chromatids will get separated. Okay. So meiosis answer is D, separation of sister chromatid. Meiosis 2 is similar to mitosis. No DNA replication before it. It separates sister chromatids. Last question, doctor.
Trisomy are best described as point where spindle fibers attach, sites of DNA replication, point of crossing over between homologous chromosomes, regions of centromere division, areas of chromosome condensation. Sir, point of crossing over between homologous chromosomes.
Yes, correct. So this is a recall question. Many times this has been asked. So answer is C, points of crossing over between the homologous chromosomes, that is the chiasmata. So that is a visible manifestation of crossing over, maintain connection between the homologous chromosome. Next question, doctor, please.
A 38-year-old woman delivers a baby with trisomy 21. The most likely meiotic error is failure of DNA replication, failure of crossing over, non-disjunction during meiosis I, failure of cytokinesis, early suppression of chromatids in mitosis. Sir, option C, non-disjunction during meiosis.
Yes, it is going to be non-disjunction that we have seen at the end. Okay, how this non-disjunction happens. So non-disjunction during meiosis I. So non-disjunction is the failure of chromosome separation. Most commonly occurs in meiosis I. It leads to either trisomy, that is like Down syndrome, or monosomy, which is a Turner syndrome. Last question, doctor.
Which of the following is the most important function of meiosis? Increase cell numbers, repairs damaged tissues, maintains genetic stability, generates genetic variation, produces identical daughter cells. Meiosis generates genetic variation. Option D.
Uh, uh, Actually, C and D are a bit confusing.
Confusing.
Maintain genetic stability if we think from the fertilization point of view, because
Point of
there is going to be meiosis, we will get haploid cells, but after fertilization, there will be genetic stability. Okay. So for genetic stability from future point of view, the option C appears correct. But current, once we think only for the meiosis, then that is the importance of genetic variation. I hope you are getting what I mean to say. Okay.
yes.
Okay. So let us see what the answer is given here. It is the genetic variation. Okay. So meiosis ensures genetic diversity via crossing over and independent assortment. It also maintains chromosome number across generations. Okay. So just take home message is that prophase 1 is concerned with crossing over, metaphase 1 independent assortment, then anaphase 1 reduction division, meiosis 2 is same like a mitosis, and non-disjunction will lead to aneuploidy, that is trisomy and monosomy. Okay, so that finishes the meiosis also.